I'd just like to begin by acknowledging the traditional owners and custodians of the land on which we meet today and the Wurundjeri people of the Kulin Nation, and pay our respects to their elders past and present and the elders from other communities who may be here today. Okay, so today it's my great pleasure to introduce Swarna. Swarna is one of our first PhD students in the lab. Started a few years ago now in the inflammation division here at WeHi. She's achieved a lot during her PhD. We've still got a good 6 months to go, but up until now it's been a real roller coaster, I think, for Swarna experimentally with the ups and downs. But I think what you'll see today is a story that's really a testament to her determination and motivation to take something through from the very beginning to the very end. And I think it's a bit sad that the universities now are cutting down on the time to finish PhDs, particularly because it doesn't really allow students to see their stories come to full fruition and publication. But I'll see what you'll see today. Swarna, she's really taken a story that starts with mass spectrometry and biochemical analysis all the way through to the generation of a novel mutant knock-in mouse. And we're now just beginning, hopefully she'll be able to finish off the in vivo characterization before she finishes. So she's done a lot of things I mentioned. She actually recently was part of a team that won this UniMelb's startup competition, coming up with novel ideas. And as part of that, she got flown off to San Francisco and Silicon Valley to pitch those to investors. She's also recently come back from her postdoc tour. And I've heard, I wasn't there, but I've heard that her talk there generated, at one of the Keystone meetings, generated a robust debate amongst some of the leaders in the field. And I think that's testament to the quality of science that she's doing. And subsequent to that, I got some emails from various lab heads overseas in the US, very interested in offering her postdoc positions. So I think that it bears well for your future career. So without further ado, I'd like to let Swarna up and present her work from her PhD. Thank you.
Thanks, James. Good afternoon, everyone, and thanks for the kind introduction, James. So during my PhD, I was interested in understanding the innate immune signaling pathway, and I worked a few projects around that. And this project, post-translational regulation of interleukin-1β, gave me the most interesting data. So all the data that I'm presenting today is part of this project. The cells in our body are constantly being, from time to time, being stressed with these pathogenic threats, and they are recognized by these receptors called as pattern recognition receptors. There's few types of these pattern recognition receptors. The one I've shown here are the toll-like receptors onto the top left, and they can sense different parts of the bacterial components, either in the extracellular space or in the endosome. And then there's not-like receptors, the NLRs. As I've shown here, peptidoglycan could then activate these receptors or sensors, and also these NLRs can form complexes called as inflammasomes. Apart from TLRs and NLRs, there are then CLRs, which is C-type lectin receptors, which are important for antifungal immunity. And then there's RLRs, which is Regai-like receptors. And apart from all these four, there's also double-stranded DNA sensors. And the most well-studied one is the cGAS STING pathway. So most of these receptors, apart from these four double-stranded DNA sensors, can all induce interleukin-1β. And therefore, interleukin-1β is very important for clearing pathogens. However, when these pathways go dysregulated and there is excess of secretion of interleukin-1β, then that results in autoinflammatory diseases. And therefore, blocking these cytokine is one of the common way of treatment in these diseases. And as I've shown here, anakinra, which is the recombinant human IL-1 receptor antagonist, was approved for rheumatoid arthritis in 2001. And canakinumab in Australia, it's been used to treat severe CAPS patients, which is chiroporin-associated periodic syndrome, which results in the severe rash, and they respond really well to the monoclonal antibody canakinumab of IL-1β. Canakinumab is also approved for SGIA, which is systemic juvenile idiopathic arthritis. And the most interesting trial is the CANTOS trial, which is canakinumab associated anti-inflammatory thrombosis outcome study, which was done by Novartis. So they recruited more than 10,000 patients who had prior myocardial infarctions, and they wanted to know if canakinumab was beneficial for these cardiovascular diseases, and the results were published just a few months back, and what they showed was it was indeed beneficial in these cardiovascular patients, but also they also found it benefited lung cancer patients, and now they're filing next clinical trials to do that. However, at the same time, there was this company which came up with this allosteric monoclonal IL-1β antibody called as ZOMAS. They was part of the UC Berkeley, and they trialed anti-IL-1β therapy in type 2 diabetes. Novartis was having its eye on this patent for a very long time, as they were the leaders in this blocking therapy, and they recently bought the patent for more than $34 million, which shows that this whole IL-1β blocking therapy is hot in pharma, and therefore understanding how the secretion is regulated is important to understand to treat these diseases. So what we know about the signaling is IL-1β, for the secretion to occur, we need two signals. The signal 1, which is the priming signal, that is given by pathogen-associated molecular patterns that I just talked about in my first slide. They activate the toll-like receptors and the MIghty88 pathway, which would then allow the nuclear translocation of the NF-κB transcription factors P50 and P65 into the nucleus and induce the transcription of pro-IL-1β and pro-IL-18 pro-inflammatory genes. The signal 2 is given by damage-associated molecular patterns called as DAMs. This could be anything from ADP, nitrogen, just a few I've mentioned, but there's uric acid crystals or cholesterol crystals which could also activate this pathway. And then it activates the inflammasome. Here I've just shown the NALP3 inflammasome because the hyperactivation of NALP3 is what causes excess of IL-1β secretion, so it's physiologically relevant. But also, this is the stimulus that I've used in most of my experiments, so I've just shown that here. So NALP3 then recruits the adapter protein ASC, which then recruits the protease caspase-1, and on this complex formation called as inflammasomes, they then get activated to then cleave the pro-IL-1β and pro-IL-18 to its active form and secrete it out of the cell. Also recently, Feng Xiao and Vishwa Dixit, they showed that caspase-1 can cleave gas-termin D, and these N-terminal gas-termin D moves to the membrane of the cells, and they cause this lytic form of cell death called as pyroptosis. This is very similar to necroptosis, where the N-terminal MLKL goes to the membrane and punctures pores. So before I talk about the ubiquitination of inflammasomes, just to give a brief introduction of what post-translational modifications are. These modifications alter the properties of the protein by adding one or more chemical groups to the amino acid residues of a protein, and they alter the properties. There's more than 200 different kind of post-translational modifications. The ones that I'm studying in my project are phosphorylation and ubiquitination. So this is again the NALP3 inflammasome, the one that I showed before, but here I've shown the different domains of this inflammasome. So the leucine-rich repeat domain is part of the NALP3, and then there's the oligomerization domain. The parent domain is what NALP3 uses to recruit the ASC protein into this complex. And then there's, through the CARD domain interaction of the ASC, it recruits the caspase-1 and activates the caspase-1, which then induces the secretion of IL-1β and IL-18. The first paper that came studying the ubiquitination in the context of inflammasomes was this paper, BRCC3, which is a D-ubiquitinase enzyme that de-ubiquitinates the NALP3 at the leucine-rich repeat domain and therefore causes its activation. Soon after that, there was this paper where it was shown HOIL1, which is part of the linear ubiquitin complex, was required for the ubiquitination of ASC, but also HOIL1 was required for the interaction between NALB3 and the ASC protein. And then this paper from the Kanagati group, they showed that sharpen, which is also part of the Lubac linear ubiquitin complex, was important for the priming step, which is the signal 1, and not for the activation step. And after these papers, there's almost 4 different E3 ligases now being described in the literature for NALB3 and so on. And this literature is rapidly expanding. And therefore, this shows how important ubiquitination is regulating this complex. And this leads me to talk about my hypothesis, which is ubiquitination is a key regulatory mechanism, which is essential for controlling R1β secretion. Also, the aims of my project was to examine and define the ubiquitination of endogenous pro-IL-1β and to examine the role of ubiquitination in activation and secretion of interleukin-1β. So just giving you a brief introduction of how this ubiquitin system works, there is deubiquitinase enzymes, which cleaves the polyubiquitin chains in the cells and creates this ubiquitin monomers. And once these monomers are free to interact, they interact with this E1 activating enzyme. This is the activation step. And the second step is the conjugation step, where the E1 enzyme then shuffles the ubiquitin to the E2 enzyme. And the third step is the ligation step, where the E3 ligase enzyme adds the ubiquitin onto the substrate through the lysine residue. So the ubiquitination happens only commonly at the lysine residue. And once there's one monoubiquitination of the substrate, then the other ubiquitins can come and tag onto the lysine residues of the ubiquitin as well, causing this polyubiquitin chains. Again, these deubiquitinase enzymes can recognize these polyubiquitin chains and cleave the ubiquitins, and the monomers are again free to go into the cycle. Now there's 76 amino acids in the ubiquitin, and these are all these other lysines that's present in the ubiquitin protein. The most well-characterized ubiquitin linkages in the literature is the K63 and K48. K63 tags onto a protein means the protein is then sent through cell signaling events, whereas when there's a K48 tag, then the protein goes through proteasomal degradation. And then there is all these specific dubs, which specific to these linkages, which can remove these linkages and make the protein to go into other events. So it's a very tightly regulated mechanism. And apart from, this is only a simple schematic, but there could be different kinds of ubiquitin chains formed. There's mixed ubiquitin chains where K48 and K63 can be having chains together. But this is just a simple overview. So I started by wanting to know that if the ubiquitination plays an important role in the NALB3 activation and R1β secretion. So for this, I took bone marrow-derived macrophages, and then I treated them with NALB3 inflammasome stimulus, LPS with ATP or LPS with alum. And then I used these deubiquitinase inhibitors to see if they had any effect in R1β secretion. So I took the supernatants from the cells after activating them, and then I measured the R1β concentration by Eliza. As you can see, when I give only the signal 1, which is the priming signal, I do not see any secretion, as there's no R1 active form being secreted. However, when I give the NALB3 stimulation with the canonical NALB3 stimulus ATP or with alum, I can see the wild-type cells secreting R1β. And in the presence of these JAB inhibitors, which are chemical inhibitors, I can see the secretion of R1β is reduced in a dose-dependent manner. Also, when I use another deubiquitinase inhibitor, the WP1130, I can again see similarly the deubiquitinase inhibitor reduces the secretion of R1β in response to NALB3 activation. So this showed that ubiquitination may regulate the NALB3 activation and R1 secretion. But we wanted to see in an endogenous physiologically relevant cell line like macrophages, we wanted to see if the ubiquitination was really happening in that cell type. So for that, we used a system called as tubes, which is Tandem Ubiquitin Binding Entities. These tubes have ubiquitin binding domains which specifically binds to only polyubiquinate protein, and we can use that to purify polyubiquinate protein in our system. But also the wonderful advantage of using the system is that we completely do not have any kind of artifacts that we would see if we use 293 T cells, which is what the literature in ubiquitination commonly you can see use. And also, the system protects against proteasome and deubiquitinase cleaving these ubiquitin chains. So I started these experiments by having bone marrow-derived macrophages. I treated them only with the signal 1, the priming signal, LPS, and then used the tubes to purify the polyubiquitinated proteins. And these proteins, these tubes are usually agarose beads with the ubiquitin domains. And once I purified them, I run the Western blot to see the proteins if they are actually ubiquitinated. So the first question that I wanted to ask was if the NUL3 components and IL-1β were ubiquitinated. So for this assay, as I explained earlier, I gave only the LPS in the signal 1, which would induce only the transduction of pro-IL-1β. And when you see the ubiquitin blot, so the top panels are my tube purifications, the bottom ones are my lysates. This is just a loading control. So when you look at the ubiquitin blot, you can clearly see with the treatment of LPS at 3 hours and 6 hours and 8 hours, the total ubiquitin protein that were purified was similar across all these conditions. And also, I could see the total ubiquitin protein in the untreated lane, but in the agarose control, there was no ubiquitin seen, which shows how specific the system is. But also, when I looked into the NUL3 different components and also IL-1β, again, the bottom panels are all my lysates, and the top panel is my tube purifications. When we look at a tube purification of NUL3, the ubiquitination of NUL3 is seen at 3 hours, and it increases with 6 hours and 8 hour treatment of LPS. And also, IL-1β showed a very similar pattern with NUL3. This ubiquitination at 3 hours, and at 6 or 8 hours, you can see a clear ubiquitination smear coming up. Also, we saw that ASC and caspase-1, there was no ubiquitination happening in this signal. So then we wanted to know if this was only specific for TLR4 ligand LPS, what if we activate with other TLR ligands like pantreases? So we went and did that. We also wanted to know what is the status of IL-1β ubiquitination when you activate the NUL3 inflammasome. So for that, this experiment, I gave the signal 2. So I activated them either with ATP or with nitrogen at different time points. And we look at the tube purification of the NUL3. You can see at the 8 hours with the pantreasis treatment, the ubiquitin smear is similar to the 8 hours LPS treatment in the NUL3 plot, showing that both TLR4, TLR1 and 2 ligand can ubiquitinate NUL3. Also, when we activate these inflammasome with the ATP and nitrogen, you can see that NUL3 is deubiquitinated, which is consistent with the literature published that NUL3 has to be deubiquitinated in order to be activated. And in IL-1β, again, you can see that at 8 hour time point with pantreasis, there's ubiquitination of IL-1β very similar to the 8 hour treatment with LPS. And when we activate the inflammasome with ATP at 20 minutes, we see an altered ubiquitination. There's much more ubiquitination smear with IL-1β. And at later time points, I see a reduction in this ubiquitination pattern. And this, probably even that, both of in the later time points, it could be just that cells dying, and that's why I'm seeing that reduced ubiquitination. So this brings me to the summary of my first part of the talk, which is the endogenous IL-1β and NUL3 are ubiquitinated in response to both TLR4 ligand, LPS, and TLR1 and 2 ligand pantreasis. And the other inflammasome components, ASC and caspase-1, are not ubiquitinated. And IL-1 has an altered ubiquitination in response to NUL3 activation. So once we knew that the IL-1 was ubiquitinated, we then wanted to know what is the function of this ubiquitination, whether this ubiquitination was tagging IL-1β for its proteasomal degradation events, or was it tagging the protein for cell signaling events. So for that, I again took bone marrow-derived macrophages, and this time I treated them with the proteasomal degradation inhibitor MG132. And as you can clearly see, in these wild-type macrophages, when I treat them, so I stimulate the cells with LPS so that I get the IL-1β ubiquitination that I usually see. And when I treat them with MG132, there's a massive increase in this ubiquitination smear. Also, it was nice to see the DUB inhibitors that I used in my previous experiments, they also increased the ubiquitination of R1β. So this indicates that with MG132, I can see an increase in ubiquitination, and that means R1β is targeted for proteasomal degradation and regulated by the proteasome. Also, this indicates that R1β might have ubiquitin K48 linkages. Also, we wanted to know what is the stability of R1β. So for this, I took 293T cells, and I transfected them with the wild-type R1β construct. And then I did a cyclohexamide chase assay, which is cyclohexamide is a protein synthesis inhibitor. And as you can clearly see, in a very short time, at 6 hours, I almost see a rapid reduction in R1β stability, showing that R1β is a short-lived protein. So now we knew that probably it's regulated by a proteasome, and it's a very short-lived protein. Now we want to know what are the residues that are actually causing this ubiquitination. So for this, we did a mass spec analysis. I took, again, 293T cells, transfected them with R1β flag construct, and then trypsinized the bands that I saw. And Laura Dagley in Andrew Webb's lab helped me to run all these mass spec analysis. So when we did a ubiquitin plot, the real nice ubiquitin smear that we saw was with this band where R1β with MG132 treatment. And on cypro staining, that was this particular lane. So we took all the bands from this lane, trypsinized them, and then Laura ran through the mass spec to see if we can see any modifications. So usually when we're looking for ubiquitination, when we trypsinize the protein, what we're looking for is the gly-gly linkage signature, showing that that is where the ubiquitination is present in that protein. And we did see that at the Lysine 133 residue for R1β. Also, interestingly, we saw a phosphorylation site next to the Lysine site, which was serine 134, was phosphorylated. And that lysine residue was conserved across many species, and the phosphor site, the serine site, 134, was not conserved in the rat. If you want to see in the whole sequence, the 2 lysine residues are present here, as I've indicated in the red. And the green fragment is the mature fragment. So caspase-1 cleaves at the aspartic acid residue 117 to produce this mature fragment. And on a protein map, the lysine and the serine residues in this linker region. And Agulia in structural biology lab helped me to do this modeling, and we found the serine and lysine residues next to each other, and they're just present in this linker region just at the very beginning. So now we want to know what is the function of this, whether these lysine residues had any functional consequences in secretion and so on. So we created mutant versions of this lysine residue, the phosphomutant, the phosphomimetic. I did all these by site-directed mutagenesis. And once we got this construct, I took the lysine mutant and I co-transfected with the ubiquitin MIC construct and in 293T cells, and did a MIC IP and looked at Western for the ubiquitination pattern. And what you can clearly see is this is my MIC IP. The K133A construct definitely showed a reduced reduction in the ubiquitination smear compared to the wild type. And these are just my ubiquitin controls. So this mutant had an effect on the ubiquitination of R1β. And then we want to know if the mutants also had an effect on the stability. So again, I did a very similar cyclohexamide chase assay in 293T cells with all the different mutants that I had. And as you can see, that I showed, like I previously showed, R1β wild type construct, it's a very short-lived protein, so I can pretty much see it degraded at 9 hard time point in the cyclohexamide chase assay, whereas the Lysine 133A construct, that mutant completely can stabilize the protein much more better than the other constructs that I have. So if we can focus on the serine phosphomutant or the phosphomimetics, the stabilization was not improved at all. So it was pretty much similar like that of the wild type, showing that this residue really didn't have any effect on the stability. It was just the Lysine mutant that had stabilizes the R1β. And also I had a bunch of different other constructs. So these constructs are the number of lysines. So there was 4 lysines between these residues, and this is 8 lysines between these residues. And this construct is 18 lysines. So this is the conserved lysines between the human and the mouse. So I took all these constructs and I also looked for stability. As you can see, I can't express this one, but the other ones really showed less stability. So this brings me to the second part, summary of my talk, which is a proteasome seems to regulate R1β, and then R1β is a short-lived protein, and R1β is ubiquitinated at the lysine 133 residue and phosphorylated at the serine 134. And the mutant lysine 133A increased the stabilization of R1β. So before I talk about how this ubiquitination can affect the secretion, the central dogma in the cell that field is this pyroptosis and R1β secretion occurs together. But recently, Kate Schroeder came up with this paper where she showed in neutrophils, the cells don't have to die in response to salmonella challenge, but they can still produce R1β. So also Steph in our lab, she showed previously that the same, that in her mast cell systems that cells don't die, but they can still produce R1β, and was shown by other papers as well. So now we wanted to see if the R1β constructs that we had any effect on the secretion. So I transfected the mutants along with the caspase-1 shown here, and then in 293T cells took the supernatants and measured the R1 concentration by Eliza. And you can see that the wild-type constructs secreted R1β. These are all the different titrations of caspase-1. And these two are the negative controls that I had, and the phosphomutants and the phosphomimetic exactly almost similar to the wild type, whereas the ubiquitin mutant was the only one which completely showed no secretion whatsoever. And this was very interesting. And when we plotted that on the Western, we can again see that with caspase-1, that the other mutants are being able to be cleaved, like that of the wild type, but only the mutant is not being able to be cleaved. And when you look at the secretion, they're not able to be secreted, whereas the other constructs can secrete R1β compared to that of the wild type. And this is just the control caspase-1 plot showing equivalent expression. So we also looked at cell death because we didn't know if the cells were just dying and throwing off R1β. So when we looked at cell death, there was no cells dying at any of these conditions, again showing the R1 secretion was an active process. Also, James previously has shown that caspase-8 can cleave R1β directly, and therefore we went ahead and did similar experiments with caspase-8. So I, again, transfected all these mutants that I had along with the caspase-8 construct, and then in 293T cells, took the supernatants and did the ELISA to measure the R1β concentration. And across different titrations of caspase-8 shown here, the wild-type secretion was completely normal, and the phosphomutants, phosphomimetics were similar. However, the secretion with the mutant construct was completely abrogated when I had a caspase-8 or with caspase-1. So, and again, the cells really didn't die with caspase-8 either, showing that this was, again, an active process that was happening. So this data was really interesting for us, that the mutant version did not allow any secretion of R1β, and therefore we wanted to know if this was really happening in the physiologically relevant system. So we went ahead and did a CRISPR knock-in mice. So we knocked in this lysine 133 residue to the arginine, and that is shown here. So magic facility, Andrew Q and Marco Harrell's lab helped me generate this mice, and you can see this is the mutation that we wanted, and we were able to generate a few of the mice, founder mice with these mutations. And we back-crossed them to remove any undesired mutations. All the results that I'm going to show from now on is from this mice number 6 founder line. So what I did was I took the bone marrow-derived macrophages from this founder mice and treated them with the NULB3 stimulus and wanted to know if they had any effect in the secretion of R1β compared to the wild type. So these are all the different treatments that I did with different time points, 30 minutes, 1 hour, and 2 hours. As you can see, there was really no differences in the secretion with most of the stimulus. The only difference that I saw was in these 3 conditions, which was with compound A at 7 hours or compound A with overnight treatment or just with alum. So compound A triggers caspase-8-mediated R1β secretion. So it was very interesting to see that these treatments had like really good R1β secretion, the knock-in, compared to the wild type. And we wanted to know, at that time we hypothesized that this could be because these are killing the cells more in a slower rate, and therefore we can capture that higher stabilization and these more secretion of R1β, whereas in all these other conditions, the cells were dying rapidly, and therefore we can't capture this event. However, before I go and talk about more about the secretion data, I just want to show you these were the controls for TNF. They all looked similar across the board, showing that this was all NOD3-mediated R1 secretion, via caspase-1. But we wanted to know if the stability was the same that we saw in the 293T cells or in the other, in the 293T experiments, was the similar that, and we did find that the knock-in mice in the macrophages, the stabilization of R1β was much more pronounced compared to the wild type. So this is again a cyclohexamide chase assay, but this is done in bone marrow-derived macrophages from the wild type and the knock-in. So I treated them with LPS to induce the R1β transduction, and you can see that again R1β is a short-lived protein that I saw in the 293T cells, but the stabilization with the knock-in mice was much more pronounced. And this was not just with LPS. When we used PAM-treces as well, you can again see the stabilization of the knock-in mice of pro-R1β was much more pronounced compared to the wild type. And now we wanted to know this degradation of R1β, how is it happening? Is it happening through the proteasome, or is it happening through the autophagy lysosome pathway? So again, I took bone marrow-derived macrophages, and then I treated them with PAM-treces to induce the signal for R1β, and also NOD3 requires but caspase-1 does not really necessarily require any induction, so you can see caspase-1 throughout. So what I did was, first of all, I started with looking at the cyclohexamide chase assay, and I can see the degradation of R1β, reduced stability, and then I can then rescue that degradation by adding MG132, as you can see there. But when I use the baflomycin, which is the lysosomal autophagy inhibitor, I'm not able to do that. So suggesting that in the wild-type macrophages, the R1β is regulated by the proteasome. Also in the knock-in, to start with, the stability was much higher compared to the wild-type, but they were still degraded almost at 6-hour time point. And I could rescue again with the MG132 treatment. However, with baflomycin, we can't see that increase in stability. So this suggested both in the wild type as well as in the knock-in, the proteasome is what was regulating R1β. Now we wanted to go ahead and wanted to see what is the ubiquitination status between the wild type and the knock-in. So I took the bone marrow-derived macrophages again here, and then I treated them with LPS. And in the presence of MG132 or without, it doesn't matter, the wild type and the knock-in showed exactly the same ubiquitination pattern. This is just a ubiquitin control showing that we got a total ubiquitin from all the conditions. So this suggested that the ubiquitination was similar across the wild type and the knock-in, and this makes sense because we are mutating only one residue out of the 19 lysine residues present in R1β, and if we take one, the others can still ubiquitinate R1β, so it could be just, the other one is that we haven't really identify the other lysine residues that's involved in this ubiquitination. So that will be one of the future directions of this project, to look into the other lysine residues that could be involved in the ubiquitination of R1β. And then we wanted to know what sort of lysine linkages are present in this ubiquitination. So for this, I did a high stringent IP with 4 molar urea in the lysate. And I use specific ubiquitin linkage antibodies, K48 and K63, and this is my IgG isotype control. So these are my lysates. So this is wild-type knock-in and knockout R1-beta, just my control again. And you can see in my pull-downs that there's K48 chains and K63 ubiquitin linkage chains coming up both in the wild-type as well as in the knock-in. So this is pro-R1-beta here. And this shows both the ubiquitination pattern as well as the linkage that are present between the wild type and the knock-in are exactly similar. The only difference we found till this point in the macrophages was the stabilization. And also, now we wanted to go back to secretion and test if this increased stabilization can actually cause more secretion. So for this, we wanted a cell system where they do not die. And we went ahead and wanted to test in neutrophils, because we know neutrophils don't die, in response to null-3 inflammasome activation. So I took bone marrows and I sorted for neutrophils with Ly6G high positive cells, and then I activated a null-3 inflammasome. And as you can see in this figure, with the treatment of LPS, 3/6 or with the interferon gamma with LPS to induce the levels of TLR, I can see R1-beta being induced. And in the knock-in, this is, the stability is much more increased. Again, this is consistent with my macrophage and my 293T experiment, where the knock-in did induce this increase in stabilization of R1-beta. All the other components of the inflammasome were pretty much similar across the wild type and the knock-in. But what was very interesting was to see that with nigericin, the inflammasome stimulus, either with the presence of interferon gamma or without, the wild type were able to secrete R1-beta, but that secretion in the knock-in was much more higher compared to the wild type. So this was very nice to see, and this was not just one experiment. In fact, when we looked at 4 different mice in different experiments, we can again see the knock-ins produced much more R1-beta compared to the wild type, whether it was present in the presence of interferon gamma or in its absence, showing that we were able to capture that phenomenon in the neutrophils. So with the TNF, again, all the TNF across all these conditions were similar, showing this was caspase-1 dependent null-3 activation, secreting R1-beta. And now we wanted to actually see if this mice can, what would be, how would react to inflammatory disease condition like arthritis. So Kate in our lab, who's an expert in this KBYN model, which is a sebum transfer arthritis model, she showed previously in her Nature Comms paper that when you used anti-R1-beta antibody, you can completely bring down the clinical severity of the arthritis in these mice. So it's an R1-beta dependent model, and we wanted to see what would be the arthritis levels in our knock-in mice. And this is a very preliminary experiment, but this is 7 mice in each group. And you can see, as you might expect, with higher levels of R1 secretion, they're trending towards having more severe arthritis, but it's still a very preliminary data, and we have to do more cohorts to actually finalize this experiment. But at least it's trending in what we might predict, that higher secretion leads to severe arthritis in the knock-in mice. Now, this brings me to the summary of my talk, which is R1-beta is ubiquitinated at the lysine 133 residue and phosphorylated at the serine 134. The R1-beta is a short-lived protein, and it's regulated by the proteasome in both the wild type and the knock-in macrophages. And then the knock-in mice had increased R1 stability in the neutrophils, leading to increased secretion of R1-beta in response to null-3 activator nigericin. The future directions of my project are to examine as I mentioned previously, to mention the other lysine residues involved in R1-beta ubiquitination. As we saw, the R1-beta ubiquitination was exactly similar across the wild type and the knock-in. And also, we wanted to look at the secretion and activation in response to salmonella infection. As neutrophils are very involved in infectious models, it would be interesting to see what happens in an infectious setting. And also to look at the knock-in R1-beta activation by using the MEF NF-kappa GFP reporter cell line to see if this knock-in had any effect on R1-beta activation. And also we came across E3 ligase during our mass spec analysis, and I've CRISPR'd them, and I'm testing it if it's the true E3 ligase for R1 beta. And finishing my talk with acknowledgements, I would first of all like to acknowledge my supervisors, especially James Vince and Kate Lawler, who've been such great support and given me so much encouragement throughout my PhD and really great ideas to keep me pushing through. And I really like to thank them for their support. All the present and past members of the Vince Lab who have been such great support and again providing me with incredible advice or with reagents, or with any support, a great lab to work in. And also the proteomics lab, Laura Dagley, who helped me run all the mass spec analysis, the Marco Herald's lab, Magic Facility, who gave me the CRISPR knock-in mice, all the members of my PhD committee, who gave me great advice again. And Keely was also very helpful along the process of my PhD. The protein lab for all the protein reagents, my Western blots that I ran, and Paul Baker in Masters Lab, especially for the CRISPR work that I did, which I didn't present in this talk, but he helped me with all these CRISPR lines that I did. And Shay and Alexandra, they again gave me all the DUB reagents that I require, and Agalya helped me with the structure modeling in the Gulbis lab. Stephen helped me with next-gen sequencing of all the CRISPR lines again. Akshay and Louise, they helped me with neutrophil purification, which was more, I found, as an art rather than science. And bioservices, taking care of all of my mice. And again, War Lab for reagents, inflammation division to do PhD. It's such a great group. And my initial funding source, University of Melbourne, who gave me the international scholarship, and then it got converted into an RDP scholarship once I got my residency. The Keystone Travel Scholarship that I got to present the same talk at a Keystone Cell Death Conference. Yeah, and last but not least, family and friends who've been great support throughout my PhD. Thank you. Happy to take questions.
Lots of time for questions. Managed to slow down. So, is there any questions?
So, with your Lysine mutant, it looked like, I'm not sure, you said that you thought the stability was different, but to me it looked a bit more like you had more protein to begin with. Have you actually charted the rate of loss and the half-life of the protein in your cyclohexamide experiments, given that you seem to be starting off with more protein in most of those assays? Like, is the rate different or the same relative?
I haven't really looked at the rate, but this is in macrophages, you asking if it's starting with a higher material. But I've done, like, if you see my loading and stuff, it all seems to be pretty similar. So I think the stabilization is much more pronounced, and that's why I'm seeing, but I haven't really looked into the rate, but probably I could do that, yeah, to see if it was different, yeah.
Yeah. Transcriptional inductions on the board.
Yeah, yeah, to look at the rate, yeah. We could do that, yeah.
Why did you change from alanine to arginine when it wasn't people?
Yeah, good question. So alanine was the first mutation I started with when I did my initial experiments, but the arginine mutation, there was a paper that came out when I was doing these experiments, and they said the arginine mutation was important for, they still showed the same lysine was ubiquitinated in that paper, so this was by Avrama, and they showed that this arginine mutation reduces the secretion of R1 beta, and it was important for the processing. And when I used this arginine mutation in my 292T cells, I really didn't see a difference, and we wanted to know if it was true, and that's why when we made the mice, we made the arginine mutation mice to see if we could see any changes, and actually what we predict is actually the opposite, where we see an increased stabilization of R1 beta causing increased secretion of R1 beta. So it's more controlling the stability and not the processing, as that paper described. Yeah, Theo?
Have you looked any more at what the serine phosphorylation was about?
Serine phosphorylation, it really didn't have any...
More into it?
Yeah, we couldn't really see an effect on its secretion, but there are still cross-linking between these pathways that can have an effect, but we really didn't go because we wanted to just keep it to the ubiquitination area, and yeah. But yeah, that's an interesting thing to see as well. Yeah.
This E3 ligase that you've identified, are you going to have a look if it interacts with other aspects of the inflammasome, or is it known to interact with other?
Doesn't really have known to interact. It's more described in a disease in the Japanese population. That's about it. There isn't much more described in the literature about that. Haven't seen any interactions in inflammasomes. So, might be interesting. Yeah, Akshay.
On that note, on the disease sort of note, are there any sort of polymorphisms that on disease associations with this lysine and leading to maybe increased secretion as a mechanism for?
Yeah, we had a look through that. Fiona helped me to do that because she has the autoinflammatory disease registry in Australia. We really didn't see any associated polymorphisms, and you don't see usually in any pro-inflammatory cytokines. The only one is the autoinflammatory cytokine IL-10, the anti-inflammatory cytokine IL-10 that has these kind of polymorphisms. But the only mutations that you see in this complex is with NUL3. You don't see even disease mutations with ASC or caspase-1. And also we had to look through the NOMAD database to look at the healthy controls. With the Sivine mutant, there were some mutations showing that it might not be really important in disease pathology, but the lysine, we didn't really see any differences in the healthy controls, showing that it might be important for survival and just, healthy pathology, I guess, yeah.
There's no further questions. Just thank you so much for a